This is Mark Giroux, CEO of Kurve Therapeutics. We formed the company to advance the standard of care for challenging central nervous system diseases by delivering drugs directly to the brain. Currently marketed intranasal technology does not reach the brain directly as the spray concentrates lower in the nasal cavity, while direct brain access is in the upper regions. VNAs, our nose-to-brain delivery platform, accesses the brain directly with controlled particle dispersion, which consists of 4 critical-to-function areas of control. Accessing the upper reaches of the nasal cavity requires precise droplet size control, atomization rate, exit velocity, and optimal turbulent flow from our electronic atomizer. The device's aerosol navigates the curved and angular paths to the top of the nasal cavity and penetrates the brain via the paths used by the olfactory nerves, thereby circumventing the blood-brain barrier. This opens a large domain of therapeutic opportunity, as virtually all of the developed drugs do not get adequate access to the brain from oral, intravenous, or other standard dosing. Kurve's VNAs platform has been used in 20 completed or ongoing placebo-controlled studies. 10 of those are peer-reviewed studies with intranasal insulin, 5 of which were in Alzheimer's disease. All of these Alzheimer's studies showed statistically significant cognition improvements versus placebo. The longest study, which ran 18 months, also showed significant placebo-controlled improvements in white matter changes and CSF neuroinflammation biomarkers, as well as favorable quality of life trends. The Kurve VNAs device is now established as a credible nose-to-brain platform and has generated the most cited research on nose-to-brain delivery of drugs to treat CNS disease. This was according to a recent review of roughly 3,000 papers on nose-to-brain delivery. The same review also reported that 2 VNAs papers were the most cited on nose-to-brain delivery of molecules larger than 1 kilodalton. While we intend to develop the current VNAs platform for Alzheimer's disease, we anticipate that further significant therapeutic advances could be possible by directing and concentrating the medication more actively once it has been delivered to the brain. Our approach does not include being in the circulatory system, rather the delivery is to the extracellular space of the brain. This status allows for the drug to be delivered and then moved to a desired location. The targeting of the hippocampus and medial temporal lobe is possible, and it is likely the most important anatomical target for moderating or reversing the progression of Alzheimer's disease. While we are getting favorable results with our current platform, we expect an amplification of efficacy from being able to increase spatial and temporal concentration of a therapeutic agent in a target area. We intend to develop a magnetic nanoparticle platform that is administered intranasally and then guided via magnets to a target area.